in chapter 5, we are going to look at 3 processes that cells go through. One of those processes is the cell division. A second process is called mitosis, and the third process is meiosis. This video is going to focus on cell division. So, cell division is the process in which one cell divides to form 2 daughter cells. And this happens in both prokaryote and eukaryote cells. However, there are some differences. It is much simpler in prokaryotes. And this is because they have a single circular chromosome. It's also because they do not have a nucleus, and they have very few other organelles. There's just maybe a couple ribosomes in prokaryotes. Whereas eukaryotes have many chromosomes. They also have a nucleus that needs to divide, and they have a bunch of other organelles like the mitochondria. They have ribosomes, they have Golgi apparatus. So there's a bunch of other organelles that have to duplicate and also divide. So in prokaryotes, cell division is often called binary fission. This is a type of cell division that occurs in prokaryote cells. This is when you have a parent cell and it divides into 2 identical, and we call them daughter cells. And so there's kind of 3 steps to cell division. The first step is the DNA replication, and this is a process of copying DNA. And so this is copying DNA prior to cell division. So this DNA has to happen, or this DNA replication has to happen before the cell divides. And this results in 2 identical chromosomes. Each daughter cell will receive one of the 2 chromosomes. Once the DNA has replicated, the chromosomes segregate or separate. They move to opposite ends of the cell, and we call those opposite ends the poles, kind of like the north and south pole. And then the last step is separation. This is when a new plasma membrane starts to grow into the center of the cell. The cytoplasm ends up getting split into 2. The new and original chromosomes are separated as the cells pull apart. And the result of separation is 2 genetically identical daughter cells. And so we have a couple graphics of this. In your own time, you should watch this animation of binary fission. And so this first diagram shows you your original cell in green. Then the DNA replicates, and you can see in orange the new cell membrane starting to grow, and down here in that second to last step you can see it starting to pinch inwards, and then it ends with 2 genetically identical daughter cells, each with half their membrane being new and half of it being old, indicated by the green and the orange. And if you look really closely, you can see the different colors in the DNA as well. Sometimes, in the second one here, the plasma membrane grows on the ends at the poles versus in the middle. The results, it's the same, it's just where a different place for that membrane to grow in. Our other diagram over here shows us the mother cell or the parent cell. Our cell will elongate while it replicates the DNA. Your DNA molecules are separating as that membrane starts to form and pinch in here. That membrane finally completely forms and creates those 2 identical daughter cells. So that's just a quick look at prokaryote cell division. We call that binary fission. And then when we look at cell division in eukaryotes, remember it's going to be more complex. Those chromosomes have to get replicated as well as the organelles getting duplicated. This happens in 2 steps, or 2 major steps, main steps. The first one is called mitosis. Mitosis is a multi-phase process in which the nucleus of a eukaryotic cell divides. So it's really important for you to know that mitosis is when the nucleus divides. Key points here that happen during mitosis is that the nuclear envelope breaks down and it will reform. The chromosomes are sorted and then they get separated. And this process ensures that each daughter cell receives a complete set of chromosomes, so that each daughter cell has the same number of chromosomes. The second major step is called cytokinesis. This is the same as in prokaryotic cells. This is when the cytoplasm divides into two daughter cells. So when you see the prefix cyto, that means cell, and then the ending kinesis means to cut. So we're literally cutting the cell. Our next video, 5.2, will really look at that mitosis and cytokinesis phase. The cell cycle is a repeating series of events that a cell goes through during its life. And this is the same for both prokaryotes and eukaryotes. They have the same basic steps. However, again, the prokaryotic cell cycle will be much more simple, whereas eukaryotes will be much more complex. The three main steps of the cell cycle are the growth phase, DNA synthesis, and then finally the cell division. So we're going to take a look at the eukaryotic cell cycle in a little more detail. It has two kind of major phases, the M phase, as well as interphase. And so the M phase will include both mitosis and cytokinesis. And another thing that happens here is the nucleus and then the cytoplasm divides. So when we're looking at the diagram, we're looking at this outer ring, and we can see the M phase in red here. So that would be the first main phase. And then the second phase is in this dark green called interphase. And so interphase is the stage of the cell cycle when the cell will grow. It performs routine life processes and DNA synthesis. DNA synthesis, that word's missing, sorry. The cell will also kind of prepare to divide. And so each of these phases is further broken down. So you can see that in this mitotic phase, we have these other phases, the P, M, A, T, and C phases. And then in interphase, where the dark green starts, we have the first gap phase, synthesis, and the second gap phase are all part of interphase. And so we can see details that happen in each of these. Like I said, the 5.2 video will really focus on that mitosis or mitotic phase, and so we're just going to look at interphase here. So interphase starts with the growth phase 1. Sometimes it's also referred to as gap phase 1. And so this is the portion of interphase where the cell grows rapidly. It's going to make proteins that are needed in order to replicate the DNA. And it's also going to copy some of its organelles. And this is all kind of preparation for cell division. Most cells will be found in growth phase 1. It's kind of the normal phase. This is your normal life. Nothing really out of the ordinary is going on. If a cell is going to start to replicate and divide, then it will enter into the S phase or synthesis phase. This is a portion of interphase where the cell's DNA is copied. And so it might be worth remembering that the word synthesis means to make. So in this synthesis phase, we're making a DNA copy. After our S phase, we have growth phase, or also gap phase 2. This is the portion of interphase where the cell will make its final preparations before it divides. It's going to make additional proteins. It's going to replicate the last few organelles. It's really just making sure that the cell is ready to divide. So the cell cycle is controlled. If it's not controlled, some bad things can happen. And so it gets controlled by these things called regulatory proteins. And these regulatory proteins control the cell cycle by signaling the cell to either start or delay the next phase of the cell cycle. I kind of think of these regulatory proteins as like stoplights. So either green light, go ahead, move on to the next phase. Maybe a yellow light, no, you better go back and fix some things. Or a red light as in, nope, abort mission, don't continue. And so these regulatory proteins make sure that the cell completes the previous phase. And not just complete it, but complete it correctly. And so there are 3 checkpoints where these regulatory proteins come into play. We have the G1 checkpoint, the S checkpoint, and then the mitotic spindle checkpoint. And so this diagram shows you where all 3 of these checkpoints fall in the cell cycle. So the first checkpoint is the G1, the cell growth checkpoint, and notice how it's ending here at the end of that first gap phase. This is going to check whether the cell is big enough and if it has made enough proper proteins for the synthesis phase. If the cell is not ready to go into the synthesis phase, it's going to loop back through this resting phase and then re-enter the first gap phase. And then it could hit this checkpoint again, and this checkpoint could say, yep, you can proceed, or nope, go back into that resting phase and try again. And so that's why a lot of the cells spend the majority of their time in this first gap or G1 phase, because they kind of get stuck in this resting loop, that there's no need to move on because they haven't built enough proteins. So if the cell gets the okay from this cell growth checkpoint or the G1 checkpoint, the cell enters the synthesis phase. And so remember, that's when we're duplicating our DNA. And so this checkpoint occurs and checks that the DNA has been replicated correctly. If the DNA has been replicated correctly, the cell will continue on to the mitosis phase. If it hasn't, the cell cycle will be aborted, and most likely the cell will die at this point. So after the synthesis phase, the cell will proceed into the second gap phase, and notice there's no checkpoint up there. Our next checkpoint comes in mitosis, and this checks whether all the chromosomes are attached correctly. This happens in the M phase in mitosis, and then the cell will continue to divide if this checkpoint is passed. So the big thing here is you're going to want to know where all 3 of these checkpoints are located within the cell cycle. So what happens if the cell cycle isn't regulated? And the main answer here is that we will see cancer start to form. So cancer is a disease that occurs when the cell cycle is no longer regulated. Those regulatory proteins are not functioning the way it should. And then the cell begins to replicate and divide out of control. So we're getting a bunch of new cells forming, and they're forming very rapidly, and usually because the cell's DNA has become damaged. And this damage could come from exposure to hazards such as radiation or toxic chemicals. And so now we call these cells cancerous cells, and they tend to divide much faster than normal cells because there's no regulation. And so this usually allows a tumor to develop, and this is a mass of abnormal cells, and you can see an example of a cancerous tumor here. And these tumors tend to take up the nutrients and space that the normal cells would need, and so they tend to kind of choke out the normal and healthy cells. It's a competition, so to speak, and the cancer cells are taking and winning all these nutrients. And so this can end up damaging tissues and organs and could eventually lead to death. So the cell cycle is a very important part of our cell's livelihood, and it's controlled by these regulatory proteins.